now we're starting to talk about chapter 15, extreme and unusual habitats. And the front part of this, the top one is Lake Frickel, and this is in the dry valleys of Antarctica. And this is a case where in the short summer, the sun can hit the glaciers and melt them a little bit, and the water runs off and it gets down in these lakes. And then the lakes, though, always have ice on the surface of them. So they're perpetually ice covered. This is essentially super dry, deserty kind of thing, but the snow builds up and the ice melts. And Mona Lake is a hypersaline lake in California. And this, these pillars of salt form because as the lake, partially because the lake has dropped down in its level, but as the water moves up through capillary action into there and then dries out, it leaves behind salt, and then that continues to go. Can anybody tell me how this might be related to Lot's wife?
And she looked at Sodom and Gomorrah, she turned into a pillar of salt.
Yeah, so when they were leaving, the husband was not supposed to look back. Yeah, and so she turned into a pillar. Yeah, she turned into a pillar of salt because she looked back. And those pillars of salt still exist on that lake because they form physically. Not necessarily that that's a true story or not, but they do exist, and that's where that idea came from. Okay, so we're going to talk about adaptations in general to extremes, and then we'll go through a series of different habitats: saline lakes, which we just talked about a little bit, hot springs, cold habitats, very cold habitats like that lake that we saw on the top, intermittent waters, ultra oligotrophic, that's an extreme environment, and deep subsurface habitats, and then at the surface of the water, neuston. So one of the first things we can talk about is how organisms adapt to extremes of temperature. And we already talked about this a little bit because when we talked about ribosomal RNA and the rate of change and the need for stability, we had a ribosomal RNA molecule up there, an image of a ribosomal RNA molecule that was more stable, had to be more stable at higher temperatures because it was a thermophilic, a high temperature loving microbe. So all these compounds basically have to be stabilized. Life is an interesting thing because a lot of it is sort of somewhere between liquid and solid. So we have a membrane, it can't really be completely liquid or it would fall apart, but also it can't be too cold because it'll freeze basically and it won't work. Proteins won't be floating around in there and the transport won't work across it and will keep the cell. So you have to have the right degree of order and disorder if you want to think about it that way. So DNA, you have to be able to get at it and then open it up and read it, and you have to be able to reproduce it. If you have DNA under really high temperatures, it will denature and the organism will die. So you have to stabilize the DNA against high temperatures if you're going to be thermophilic, and you do this by increasing the number of CG pairs. Anybody have any idea why the CG pairs are more stable? Has to do with hydrogen bonding, right? Good, 3 hydrogen bonds exactly, as opposed to 2. Good, Cassie and Gary got that. Right, so there's more CG pairs, and then also there's proteins that are around DNA that can stabilize it. So there's more proteins to stabilize it. Ribosomal RNA and messenger RNA has a little introductory part that has to have a particular conformation. They have more extensive areas of base pairing and areas that need to hold conformation. Membranes, higher temperature, you increase saturation, branching, and length. And the archaea have a very unique ether lipids that basically cross-link inside of the cell membrane. this makes sense. So if you think about why it is that butter is solid, um, but vegetable oil is not solid at room temperature, right? So what temperature does butter become liquid? Said differently, if you take a stick of butter that's at room temperature and you put it in your hands, hold it like this, what happens to it? It melts, right? Okay, so butter is a form of animal fat. So to work, it has to be at, we're warm, right? So it'll be liquid and work in membranes if if it's in an organism that's warm. So why is it that plant lipids are liquid at room temperature? They're unsaturated. That's correct. But why? What's the difference with the plant? Not less fat. Okay, so plants still have cells. They still need to have cell membranes, right? But like today, outside, what would happen to the membranes if they were made out of lipids such as butter or some of ours? They don't need to have it warm for them to use. Yeah, that's almost there. Not only that, but if they do get cold enough that they solidify, then they can't be growing, right? They can't be active. Their cells can't be active. They need to deal with cold temperatures. So you can imagine how even if, let's say, a fish that lives in cold water would have membranes that are more fluid at low temperatures than one that always is found in hot water, because they need to keep their membranes at that level. So these are adaptations to high temperatures, right? But you can flip it. The opposite adaptations happens to low temperatures. So at low temperature, DNA would have fewer binding proteins, fewer CG pairs. RNA would have less extensive regions of base pairing. Membranes would be unsaturated. They would be less branched and shorter, the lipids and the membranes. And the archaea would have to not link their lipids so much if they grow in cold water. Oh, and I'm actually asking the question, why are fats from plants and fishes more healthy for people than those from warm-blooded animals, such as butter or beef fat? Right, it's just because they have lower melting points, so they're less likely to deposit on your arteries. There's more adaptations to extremes. Proteins also have to be thermally stable. Remember, protein works by jiggling, right? But it has to maintain its active site. So it has an active site that's the right shape for something to fit in. But once it fits in there, it's either going to be making bonds or breaking bonds, and it does that through thermal activity. So it's got, again, it's in that balance between being too hot and falling apart and denaturing, or being too cold and not moving enough to actually make the reaction happen. To do that, they have more beta sheets and alpha helix form. They tend to have, at higher temperatures, they have more hydrophobic cores, and they have a higher external charge. So all those things are what give proteins their three-dimensional shape. In addition, there's other extremes other than temperature. Salt water. In highest salt, water essentially becomes limiting at hypersaline environments. But there's some problems with it. Organisms need a way to stabilize proteins if there's too many ionic interactions. So we talk about just above here how this high external charge stabilizes proteins, right? If you have too many salts, it interferes with the ability of the protein to basically attract itself. And so you've got to stabilize your protein against those ionic interactions. So complex animals just pump out salt and just keep regulate salt at the right level. But there's some halophilic bacteria and archaea that are just isotonic with the water, that they conform to the salt that they're in. And then they have to have specialized ways to deal with the salt destabilizing their systems. Some organisms will use glycerol to remain isotonic. So they'll produce glycerol so they don't burst when they get, they don't collapse when they get in high salt environments, right? The water doesn't want to rush in and then burst them if they try to keep themselves far from being isotonic. Lack of water is similar to salt because organisms in temporary habitat have to be able to withstand desiccation. And when you desiccate, you basically get saltier and saltier if you want. Water becomes more limiting.